Bio 200 Lab, and this is my presentation on the gut microbiome. So to start, microbiome is the types and proportions of microorganisms living in a given location. In this lab in particular, we're looking inside the intestine or gut microbiome of an animal. For many animals, like humans, they have several microbiomes in their bodies, but we're just focusing on the gut. So, for starters, we look at this because the gut microbiome has been correlated to a variety of aspects in animal and human health. An example of this is in obesity. Some studies have shown that there's a difference in the gut microbiome composition for individuals if they're obese or if they're lean. And there's been a correlation between a species in a particular phylum with obesity, and then with lean individuals, they would have a smaller proportion or larger proportion of the species in that phylum. And the data from one of these studies, it seemed to be correlative rather than causal. As for most of the studies, there hasn't been really like a direct causal argument for most of these things. It's usually like a correlative kind of study. Another example of a health factor influenced by gut microbiomes is IBS, or irritable bowel syndrome. There's been some studies that a certain type of microbe was associated with some pediatric patients having IBS, and they would also have increased pain correlated with a higher abundance of that bacteria species. And that showed like a direct correlation, but yet again, not like direct causal, it's usually correlation. In our study in particular, we looked at flat-coated retrievers, which is a type of dog, and we predicted that, like the other studies before, the difference in composition of the gut microbiome in this species is going to be due to a variation of health factor. For our health factor, we picked age. So we predicted that basically the change in age is going to have a different correlation of microbiomes, like composition. Okay, so to go over the overview of our study, this is our materials and methods. For the animals we compared, we had 4 fecal samples from 2 different dogs. So basically, the fecal samples were collected 6 months apart. So basically, we had 2 different dogs, collected their fecal samples at one time, and then 6 months later, the samples were collected again to show aging. The first dog, his name was Hoshi, or her name was Hoshi. She ranged in age from 1 year old to 1.5 years old. Our second dog, Nova, she ranged in age from 5 to 5.5 years old. So basically, we show a 6 months difference. For each of these dogs, they were both females. They both weighed around the same weight, 50 pounds. They were in very good health with no known health issues. So after 6 months, both dogs also no longer consume supplements of these certain type of biscuits, table scraps, yogurt, and they both switched their food from dry food to regular food. But in particular, the only real difference was that Nova's exercise changed from extensive to regular after 6 months. Basically, these dogs have like the same exact health factors. They have the same exact diet. Really, the only thing that's different between them is their age. So overall, to study and actually find the composition of the gut microbiome, there's 3 major steps. First, you have to isolate the DNA from the fecal sample using a commercially available kit. And then after that, you have polymerized chain reactions, or PCR reactions. Those are done to amplify that particular region that we isolated. And after that, the amplified regions of those DNA is then going to be quantified, and they're going to be tagged with a particular index tag. And then after that, the third and final step is going to be determining the nucleotide sequence of those amplified regions using a next-gen sequencer. So to get in more particular into our steps, focusing on the purification and concentration of DNA, we use to isolate the DNA from fecal matter, we used a commercially available kit from QIAGEN Incorporated. It was called the QIAGEN kit. So basically, we use that to purify DNA. It's basically based on, you put the stool sample in a buffer, prevents enzymes from degrading the DNA, and the DNA binds to a certain filter, and it goes through a column, and basically you just wash away the impurities, and that allows you to isolate DNA. That purified DNA was then concentrated 10 times using another kit called Zymo Kit from Zymo Incorporated. This basically concentrated the DNA 10 times. So an increase in concentration tenfold. Okay, and it was basically the same exact thing, like put it in a filter, wash away the impurities, and then you'd have a concentrated, isolated DNA sample. So then in order to check if this was right, we used absorption spectrophotometry to determine the concentration of our DNA, and we wanted to see if there was any contaminating proteins or RNA in the sample. So it was done using the nanodrop spectrophotometer, and we did not just check the concentration, we checked absorbance values at, basically, so we checked it at, the absorbance is 260 nanometers, and then we wanted to put that in comparison to 260 over 280, we wanted to see that ratio, and we checked the concentration after that, and with that we were given the volume. We could do that, and we could find the volume for 100 milligrams of that sample that we want, and so that we could do a proper dilution for our sample. So this allowed us to determine, obviously, if the dilution was required. So once we knew that the concentration of the DNA we performed was good, we performed amplicon PCR to amplify the regions of the 16S ribosomal RNA gene. Specifically, we're going to amplify the V3 and V4 regions. We chose this because it's a particular part in the RNA gene that's particular to bacteria samples. And so basically, with the V3 and V4 regions, those vary between different bacteria, so we could check those regions and check them to like a database to see what the actual composition of bacteria was in our sample. So after we amplified that, we then went to gel electrophoresis, and basically the gel electrophoresis separated DNA fragments of different sizes using an agarose gel, and basically what it did was check to make sure that we amplified our DNA properly, and in particular our gel was set to 250 volts because that was what it's what the procedures said. Just want to make a side note also, all the procedures were done in accordance to the manufacturer's instructions, and we figured it out because that was what the lab manual told us to do. So after we checked our results and they were right, we did PCR cleanup reactions. Basically, these PCR cleanup reactions were done to clean up and remove the components of the amplification buffer. It was it was a bunch of different chemicals that were in the way of the actual DNA component, but we needed to use them as a buffer for amplification PCR. And we also had forward and reverse primers. Those were removed as well with the cleanup reactions. And this allowed us to actually perform, this was really crucial in allowing us to perform our actual indexing PCR reactions, where you have to tag the DNA sequences. Now these indexing PCR reactions, like I just said, they added a unique set of short sequence tags to the ends of the amplicons for each of the samples with indexing PCR reactions. So basically it's like two tags at the beginning and end of the RNA sequence. And these are done because we need a way to tell which sequences came from which samples, since all of our DNA sequencing is done in a large mixture that combined all of our classes of samples together. So when we sequence the DNA, we'll know which our sequence came from because we have those tags on them. So after our indexing reaction, we cleaned it up again just to remove the same components as the last reaction, like the buffer, the forward and reverse primers. And afterwards, we used a fluorometric assay to determine the concentration of our amplified sample to make sure that we were right and had everything indexed, and we could actually even sequence our results. And we use the fluorometric assay because it's much more accurate than a spectrophotometer. They're much more accurate readings overall. And the DNA, after that, we did the assay and checked that it was right. We diluted it to prepare it for sequencing. And then finally, we used the next-gen sequencer to determine our nucleotide sequence with a DNA analyzer. Okay, so after we got all of our methods and materials, after we finished our reactions and all that, we looked at our results. So basically our results, these are the table for the nanodrops, all the concentrations, our PCR calculations. We had we tracked all the volumes for the reagents of the PCR reactions, like master mix, DNA, control DNA, and DNA that's just water. We also had a positive and negative control on this. Our positive control was just a sample of pure E. coli DNA, and the negative control was just water. Okay, so afterwards, and then the gel electrophoresis, basically what we have is, this is our group side of the thing, and we have our base pair length. It goes from 100 to 4,000. All of our samples found, they showed readings between like 500 and 800, which is what we want because it was between, we wanted around 550. And for our, the indexing reactions, we have our chart of our fluorometric readings and our calculations to make sure, our calculations for the actual dilutions themselves. But as far as sequencing goes, our samples, we couldn't really use our samples because they weren't done yet. None of the samples in our class were actually done, so we didn't actually get our sequencing results yet. They still haven't come in. So we had to use samples from past years